Yes, sir, I am ready.
Okay. So, very good afternoon, my dear friends. Hearty welcome to our CPGET 2024 free online class. Today's our eminent resource person, Dr.A.SHAILAJA Madam Garu. Madam is going to explain about the BRYOPHYTA. Madam has clearly explained in previous class the classification and the general characteristic features of the BRYOPHYTA. So that is also most important. Now, Madam is going to explain about the one of the most important topic in BRYOPHYTA, that is hepatic apsida type study. That is Marchantia. Okay, Madam, please start your session and share your screen, Madam. If possible, please click on slideshow, madam.
Sir,
If please click on slideshow. ఇక్కడ ఉంది చూడండి. ఈ హోమ్ ఉన్న దగ్గర ఈ ట్రాన్సిషన్స్ యానిమేషన్స్ పక్కన స్లైడ్ షో అని రాస్తుంది.
sir,
ఇక్కడ పైన వచ్చినాయి మేడం?
okay, sir.
నో నో నో.
Clear, sir. Okay, na?
రాలేదు కదా మేడం? ఓకే మేడం, నో ప్రాబ్లం. ప్లీజ్ ప్రొసీడ్.
బట్ నాకు సిస్టంలో చూపిస్తుంది సార్.
ఇక్కడ మనకు ఏంటంటే ఈ మిగతా స్లైడ్స్ కనబడుతున్నాయి అన్నమాట, పక్కన స్లైడ్స్.
ఆహా, ఓకే ఓకే.
మేడం మీరు ప్రొసీడ్ చేయండి, మీరు స్టార్ట్ చేయండి మేడం, నో ఇష్యూ. లేదంటే మళ్ళీ షేర్ చేయండి.
My dear students, good afternoon, all of you.
Good afternoon, ma'am.
Shall I start?
Yes, you can start, ma'am.
Okay. Today we are going to discuss about the hepatic apsida. The division hepatic apsida is represented by totally 280 genera and 9,500 species. Okay. But in that, the plants in this group looks like liver. So hence we are called that liverworts. Hepatic apsida members are called as liverworts. The members of hepatic apsida grow on moist soils and shady localities. So some of them also grow. Already we have discussed these points in yesterday's class, but I will remind. Okay. So that members of hepatic apsida mainly grow on moist soils and shady localities. So these some of them also grow on the surface of the water also. So example for that Rixia. Okay. So the plants are gametophytes. The plant body is either thalloid or leafy stems, as in Porella. One of the examples that is leafy stem has that in Porella we are seeing that leafy stem, and the plant body maximum thalloid, and the leaves possess middle vein, and the internal organization is well differentiated. The chloroplasts in the chlorophyllous tissue do not contain pyrenoids. Okay, so we are going to first that classification. Okay, so Engler classified the members of Hepatic A into three orders, that Marchantiales and Anthoceratales and Gengermoniales. Okay, Marchantiales, Anthoceratales and Gengermoniales. So Engler classified the members of Hepatic A into three orders. Okay, three orders. See here, this is the classification of Marchantia. That division Bryophyta, class Hepatic apsida, order Marchantiales, family Marchantiaceae, genus Marchantia. Out of these three orders, we are going to discuss today that Marchantiales order. The Marchantia belongs to order Marchantiales. Okay, division Bryophyta, class Hepaticapsida, order Marchantiales, family Marchantiaceae, genus is Marchantia. Okay, next, see here, this is a gametophyte.
Madam, please, slide is not moving, madam. Only first slide is we are observing. Only
Uh,
one slide is here.
oh, okay, sir. I will. ఎందుకలా అవుతుంది అసలు?
Okay. Now, okay, ma'am.
ఓకే మొబైల్తో కనెక్ట్ అయ్యాను ఇంకా అది క్లోజ్ చేశాను సార్ సిస్టమ్. ఓకే సో this Marchantia belongs to division Bryophyta, class Hepaticapsida, order Marchantiales, family Marchantiaceae, genus is Marchantia. Okay, so gametophyte, this habit and distribution. So the genus Marchantia consists of 65 species distributed all over the world. Okay, Marchantia polymorpha is widely distributed species. There are 11 species reported from India. Okay, first we are going to that introduction, Marchantia introduction. This is a gametophytic plant. So total Marchantia consists of 65 species distributed all over the world, but Marchantia polymorpha is widely distributed species. Okay, so totally in
Ma'am, your voice is not coming.
is okay.
Yes, madam. Proceed.
The screen is
Okay,
visible.
ma'am. Okay,
Okay,
ma'am.
okay. So this is the Marchantia name is given. This is originated based on the French scientist Nicolas Marchant. So that's why we are called Marchantia. Okay, the thallus looks like liver. Hence, this is called liverwort. Okay, so first we are going to discuss the gametophyte. So external characters. See here, gametophyte of the Marchantia thallus is liver shaped. That is, these plants are called liverworts. The gametophyte shows dichotomous branching and dorsiventral differentiation. Already in the last class, we have discussed that dorsiventral. What is the difference between that dorsiventral positions? Okay, dorsal surface is green in color due to the presence of chlorophyll and shows a midrib. Okay, so see here in this diagram, you observe that dorsiventral differentiation observed in the Marchantia. Dorsal surface is green in color due to the presence of chlorophyll. Dorsal side is photosynthetic and prepares the food material. Ventral side is colorless due to the absence of chlorophyll. That ventral surface produces smooth, unicellular and rough-walled rhizoids for absorption and scales that provide protection from the drought conditions. Okay, this is the external characters. About the thallus. The thallus is very flat and prostrate. What is the prostrate now? What is the prostrate? And we can see the prostrate and erect branches observed in the Marchantia. What is the prostrate? Naina, tell me.
Ma'am,
What is the prostrate meaning? The thallus is prostrate, flat prostrate.
ma'am, you said no, ma'am, yesterday's class, prostrate and erect.
What is the meaning of prostrate?
Uh, it is trees are snake like growing, ma'am.
Growing. Erect means?
Erect means the trees are growing straight forwards.
Straight, dichotomously branched with dorsiventral structure. Some are erect and some are prostrate. In this apex of each branch, a notch is present in which the growing point is located. Okay, one notch is there. So that is the growing point. So the thallus has a conspicuous midrib which is marked on dorsal side with a shallow groove. See here, some of the midrib is there. Okay. So the deep
Ma'am,
green dorsal.
what is the midrib?
Midrib means madhya eeni antam kada. Veins amma, vein.
Okay, ma'am.
Veins are on the leaf surface. We are observed that midrib and side veins are there. Okay. So that
Okay,
is
ma'am.
midrib means in the middle of the center point. Okay. We observe that
Okay.
the center of the leaf. That is the midrib. The thallus has a conspicuous midrib which is marked on dorsal side with a shallow groove. The deep green dorsal surface is marked into small diamond-shaped areas which indicate the outline of the air chamber with just beneath the upper epidermal layer which show opening of the air chamber. See here. This is the some areas are there. These indicates that air chamber. Okay, just beneath the upper epidermal layer. These are the air chambers. On the dorsal side, we are observed that cup-shaped structures that are present known as gemma cups. Already in the yesterday's class, just we are giving that vegetative reproduction by the gemma cups only in hepatic apsida. That very important gemma cups we are observed in the hepatic apsida only. They contain special vegetative reproductive bodies known as gemma. Okay, gemma on the ventral side, numerous scales and rhizoids are present. Ventral side rhizoids are present. Scales and rhizoids are observed, but dorsal side gemma cups are there. Okay, gemma cups. So these are the for the vegetative reproduction. Okay, helps in the vegetative reproduction. It's very important. So multicellular and one-cellular thick scales are present in 2 to 4 rows on each side of midrib. The rhizoids are useful for the absorption and anchoring to the ground. There are two types of rhizoids, smooth-walled and rough rhizoids are there. At maturity of the thallus, special upright branches are developed and these are of two types, antheridiophores and archegoniophores. See here. So this is a gemma cup. See here. Gemma cups arise on the midrib of the thallus. These are helpful in vegetative reproduction. Some stalk-like structures arise on the gametophyte plant. Stalk which bears archegonia is called archegoniophore and the stalk bearing antheridia is called antheridiophore. So some diagrams I will show you. What are the antheridiophores and what are the archegoniophores? In the yesterday's class also, I will give that clarity on that archegoniophore and antheridiophore. Okay, some stalk-like structure, same look like that umbrella-like structures. Okay, stalk-like structures arise on the gametophytic plant. Stalk which bears archegonia is called archegoniophore, and stalk bearing the antheridia, these are called antheridiophore. Okay, all are you understood? What is the antheridiophore? What is the archegoniophore? Okay.
Yeah. Yes, ma'am.
Okay. So this is the transverse section of the Marchantia thallus. It shows three main parts. See here. This is the thallus structure. Okay. Thallus structure, internal structure of thallus. So it will show three parts, main parts: epidermis, assimilatory region, and food storage region. Okay. See here. This is the epidermis. Epidermis is the divided two types of divided epidermis is there, upper epidermis and lower epidermis. Epidermis has upper and lower epidermis which have single cell thickness. Only single cell thickness తో మనకి ఇక్కడ చూడండి మీరు ఈ డయాగ్రామ్ లో అబ్జర్వ్ చేస్తే కనుక upper epidermis and lower epidermis which are only single cell thickness. Okay, so and upper epidermis has air pores which are cross shaped and useful in gaseous exchange. Okay, see here. This is the air pores. Gap on the chudandi air pores. Upper epidermis have the single cell thickness and these are has air pores which are cross shaped and useful in gaseous exchange. And air pores are absent in the lower epidermis. It develops unicellular, smooth and rough rhizoids and multicellular scales. ఇందాకనే చెప్పుకున్నాం కదా, dorsal side and ventral side. Ventral side we are observed that rhizoids, smooth and rough, two types of rhizoids are present on the ventral side. So that is lower epidermis. Okay, ఇక్కడ మనకి lower epidermis లో చూస్తున్నాం scales and smooth rhizoids are present. Okay, that is the lower epidermis, and upper epidermis having the air pores. అర్థమైందా నాన్న? ఓకేనా?
Yes, ma'am.
Okay. Next, see here, assimilatory region. Tell me.
This Marchantia and Gametophytes are same, ma'am. Marchantia different, Gametophytes are different.
In Marchantia, the thallus is called gametophyte. So that
Okay.
position, okay. In the Marchantia, we are observed two stages, gametophytic stage and sporophytic stage. Marchantia is the one of the lesson. Okay, one is the type study, belongs to
Okay.
hepatic. Okay.
Hepatic?
Hepatic
ma'am?
apsida.
Okay, ma'am.
So in last class we are observed three classes are there: hepatic apsida, anthoceratopsida, and
Bryophyta.
bryophyta. Okay. From the hepatic apsida, this is the type study, Marchantia. Okay. So in that order is Marchantiales. From the hepatic apsida, that order is hepatic apsida is class, that Marchantiales is order. Okay. From the Marchantiaceae family, Marchantia is the one of the genus. Are you understood?
Yes, ma'am.
Okay. So this is the type study. Type study ante manam em chevuthamamma? Habit, habitat, structures, and then thallus internal structure, and then go to reproduction, and what is the life cycle, heteromorphic alternation of generation. We can see in the bryophytes annam kada general characters lo. So
Yeah, yes, ma'am.
all characters ఇప్పుడు మనం ఈ టైప్ స్టడీలో చూస్తున్నాం. ఓకేనా?
Okay, ma'am.
ఇప్పుడు క్లారిటీ వచ్చిందా?
Yes, ma'am. Yes, ma'am.
Okay. So next, in the thallus structure, the second part is assimilatory region. Okay. Assimilatory region below the upper epidermis is the region is called assimilatory region. See here, this is the air chamber is there na. Second one, below the upper epidermis, what is there na? Chlorophyllous filaments, air chamber, that is called assimilatory zone. Okay, clear?
Ma'am, air chamber means what?
Air chambers means that is the gaseous exchange come from that cells. Okay. That is air pores are that is the whole from outside and inside, that gas exchange. But this is the transferring zone, air chambers. Okay. So in that assimilatory zone, air chambers, branched chlorophyllous filaments are present in the air chambers. So these chlorophyllous filaments are there. This is the air chamber means that is the place. Okay. Gases exchange come from outside. Okay. So in that air chamber, what are the cells are present? Chlorophyllous filaments are there. See here. Okay na? This is the air chamber means empty gap. So in that gap, that chlorophyllous filaments are there. Okay. So these branched chlorophyllous filaments are present in the air chamber. Third one is the storage region. See here, below the air chamber, that total region is called storage region. Just below, next to the assimilatory region is the storage region. This part is made up of parenchymatous cells. Starch grains are present in the parenchymatous cells as well as some mucilaginous cells at some spots. ఇక్కడ చూడండి మనకి mucilaginous cells. Some we are observed that some cells are mucilaginous cells and all are parenchyma cells. That storage region, all cells are storing the that starch. Okay, that is the third part. This is the thallus internal structure, Marchantia thallus internal structure. In this structure, three parts are there: epidermis, assimilatory zone, and storage region. Are you understood?
Yes, ma'am.
Okay na?
Okay, ma'am.
So next, coming to the sporophyte director. Okay. Next, after the thallus internal structure, we are going to reproduction. Okay.
Ma'am, in exam, They'll give questions about bryophytes, ma'am.
Yeah. One or two questions coming from the bryophyta.
And sporophytes also.
Sporophyte means that is the internal structure of bryophytes, na? Marchantia, Anthocyrus, and Polytrichum, internal structure. So that is one of the part in the type study. Sporophyte, Marchantia sporophyte, Anthocyrus sporophyte, and Polytrichum sporophyte. And lastly, we are going to learn that sporophytic evolution in bryophytes. That is a long question actually.
Okay, ma'am.
Very important question. So in that sporophytic evolution, we are knowing that what are the differences from one type study to another type study. Okay, what is the differences between one sporophyte to another sporophyte? Okay.
Okay, ma'am.
So next we are going to first vegetative reproduction. That gemma is very important. So gemma cup. Okay. So germination of gemma, formation of gemma. A number of shallow cup-shaped structures with hyaline dorsal surface of the thallus. These are known as gemma cups, cupules. Okay. Wait.
Okay,
Now.
ma'am.
So first I will explain about the gemma. Each gemma has a hyaline stalk with a multicellular discoid body. This is the gemma. Okay. So this is the stalk and with a multicellular discoid body with two growing points situated on either side of the lateral margin. In the gemma, majority of the cells contain chloroplasts, but some superficial cells remain colorless, and from these colorless rhizoids develop later on. Okay, this is the gemma. So this is the gemma cup. Okay, so this is the gemma cup. Below the diagram is gemma cup, and this is the gemma. Okay, so see here. A few cells possess oil bodies, and some cells from the floor of the gemma cup gives rise to mucilage cells. These cells help in the breaking away of the gemma from their stalk and the liberation of gemma. A gemma, after falling on a suitable soil, gives rise to new thalli. Okay, so this is the gemma when falling on the new soil surface, and it gives rise to germinate and give the new thalli. This is the vegetative reproduction part. Gemma cups are helping in the vegetative reproduction. Remember that Marchantia. So gemma cups are developed in which member of bryophyte అన్నప్పుడు Marchantia గుర్తుపెట్టుకోండి. ఓకేనా? That is about the gemma. So next we are coming to the sexual reproduction of Marchantia. So it is oogamous. Naina. Hello? Naina.
Ma'am.
Are you there?
I am there, ma'am. I'm there.
Okay. So this is about the vegetative reproduction. That gemma is very important. Okay.
Okay, ma'am.
So next we are coming to the sexual reproduction. So in the sexual reproduction main continuous 3 classes అయినట్టున్నాయి మీకు.
Ma'am, what, ma'am?
కంటిన్యూస్ 3 క్లాసెస్ అయినాయా? ఎందుకు డల్ గా ఉన్నట్టున్నారు? యాక్టివ్ గా లేరు ఈ రోజు అంటున్నా.
Ma'am, morning onwards online classes only, ma'am.
Today onwards.
One is yoga about yoga, International Yoga Day, and one is Pteridophyta, and one is now you are saying.
Okay, no problem. So I will tell you about the what are the important points is there in the Marchantia. So this is in the Marchantia, in the sexual reproduction, we are observing that female and male sexual organs are already in the yesterday's class discussed that antheridia and archegonia. Okay, so the antheridia are born on special stalked branches, that antheridiophore. So same like that specialized stalks in female sexual organs also, that is archegoniophore. ఏదైతే స్పెషల్ స్టాక్ లైఫ్ స్ట్రక్చర్ ఉందో అది ఫీమేల్ అండ్ మేల్ సెక్సువల్ ఆర్గాన్స్ ఎక్కడ ఫామ్ అవుతాయి అంటే ఆంథెరిడియా ఫామ్
Ma'am.
అయ్యేదేమో ఆంథెరిడియోఫోర్. చెప్పండి అన్న.
Screen is not visible, ma'am.
యాక్చువల్లీ నేను స్టాప్ చేసేస్తున్నాను అన్న, అది జెమ్మ కంప్లీట్ అయింది కాబట్టి అప్ టు మళ్ళీ 405 నుండి ఉంది అది. సో సో దానికోసం నేను స్టాప్ చేసేసిన. ఓకే, సో నెక్స్ట్ సెక్సువల్ రిప్రొడక్షన్ లో ఆంథెరిడియా అండ్ ఆర్కిగోనియా. ఎక్స్ప్లెయిన్ చేయనా కంపల్సరీ?
Ma'am, what, ma'am?
డయాగ్రామ్స్ మీకు కంపల్సరీ ఇది స్లైడ్స్ పెట్టనా? నో నీడ్ టు షేర్.
Ma'am, no need of diagrams, ma'am. I explain theory part.
Okay. So just I will give the what are the important in this.
Ma'am, we are writing
sexual reproduction.
exam about objective part only, no, ma'am.
No. Yes. Okay. So
That's why
mainly.
I'm
Okay. So what are the main important things in the sexual reproduction? So antheridia and archegonia. So you have an idea of the sexual reproduction of Marchantia. Okay. So what is the main part in the sporophyte? That is the questions coming from the sporophyte part. So I will show you. I will explain that. See here, this is a sporophyte. Okay, so from the antheridia, that antherozoids are developed, and from the archegonia, that egg cell is developed. After the fertilization, zygote is formed. Okay, so that is, next coming to the sporophyte. It's very important that this is the exam point of view is very important. Okay, sporophyte. As soon as the zygote is produced, the sporophytic stage starts. So that is the developmental stage starts from the zygote. Sporophyte development is started from zygote. Zygote formed in the venter of the archegonia. It is unicellular, uninucleate and diploid cell. So this zygote develops its cell wall, undergoes a transverse division and develops upper epibasal cell and lower hypobasal cell. See here. So this is the Marchantia sporophyte. Okay. So this is the Marchantia sporophyte. So the stalk of the archegonium begins to elongate and becomes to 3 to 4 centimeters long. The cells of the venter also divide periclinally and form calyptra. See here. So ikkada manaku chuste calyptra anedi untundi. So that is one of the part as the sporophyte. And next, a ring of the cells of the base of the venter divide and collar-like growth known as perigonium. So pseudoperianth and calyptra. See here, this is the calyptra and pericytium. So these are the walls. Okay. So after these ring of cells at the base of the venter divide to form a collar-like growth. It is known as perigonium or pseudoperianth. It grows around the archegonium and forms a covering. So this is a like that covering. Okay. So the zygote divides into upper epibasal cell and lower hypobasal cell. Zygote divides and forms this sporophyte structure. See here. So next, the second division is right angles to the first and develops a globose quadrate embryo. This is called quadrant stage. So how to that sporophyte is developed, I will explain from these points. Okay, this is called quadrant stage. So then what happened that after the quadrant stage with the longitudinal divisions, it develops into 8-celled octant stage. So the zygote formed divisions periclinally, that going to divisions undergoes a transverse division and develops two cells first. Okay, the zygote divides two cells as the upper epidermal cell, basal cell and hypobasal cell. So then the two cells are going to divide and form the quadrant stage. First four cells evaithe form avutayo, that is quadrant stage. That four cells are going to divide and form the 8 cells, that is arctant stage. So then the divisions are irregular and lead to the growth of the embryo. Later the divisions in the epibasal and hypobasal cell are not the same. So further, what is the growth of a sporophyte? It is enclosed by calyptra and pseudoperions in Marchantia. The sporophyte is completely parasitic on the gametophyte. Please remember this. In the Marchantia, the sporophyte is completely parasitic. This is a dependent on the gametophyte. Okay? Remember this. This is very, very important. Note this point. Okay, Marchantia is completely parasitic, completely dependent on the gametophyte. The sporophyte consists of three parts: food, seta, and capsule. This is the structure of Marchantia. Okay, so three parts are there: food, seta, and capsule. Okay, food, seta, and capsule. So food. This is the food. Okay. So this is the food. See here. So in the food, we are observed that the divisions taking place in the hypobasal cell lead to the development of parenchymatous cells. Cells at the basal part of this tissue develop into food. Naina. Hello?
Ma'am,
Clear on the voice clear on the okay na ardham avutunda confuse unda emana.
Madam, proceed.
Hello? Okay. So this is the food. So in the food, that cells at the basal part of this tissue develops into food. So that is the first part. Well developed a food is bulb shaped. Naina, tell me. The well developed food is bulb shaped structure. Tell me.
you asked me something. No, ma'am, I can't hear you.
Who?
Now only you called me and you asked me some
Okay,
question. No,
madam,
ma'am.
you may proceed. Naina,
Sir.
I asked you, is it clear? Am I audible? Madam has told you. You are not responded properly. Okay?
Sir, madam voice is not clear, sir.
No. Your net interruption, you are not understand. Madam, you may proceed, please.
Okay, sir.
So that in the food, that is a bulb-shaped structure. It is embedded in the gametophyte and helps in absorption of water, minerals, and nutrients. So that is the food. Okay. First part in the sporophyte structure. So next one is the seta. See here, this is the seta. So this seta is present in between the foot and capsule. So parenchymatous cells above the foot or in systematic longitudinal rows. Initially, the divisions in the cells of the seta are very less and the seta will be very short. See here, this is the middle part. The foot and in between the capsule and foot, that is the seta. Okay, so longitudinal rows of cells are present in the seta. Initially, divisions in the cells of the seta are very less, that seta will be very short. But later, at the time of spore formation in the capsule, divisions in the cells of the seta will be very rapid and results in the sudden elongation of seta, due to which the capsule tears out of the calyptra and the pseudoperions at the time of the spores releasing time. Okay, that is the seta. So next one is the capsule. So capsule, it's very, very important. The cells are derived from the epibasal cells, develop into the capsule. Okay. So in the capsule, we are observed that the periclinal divisions takes place in the cells in the middle center of the capsule. So these outer in these cells, we are observed that form outer the amphithecium and inner endothecium. Okay, some of the cells periclinal divisions takes place in that cells from the outer amphithecium and inner endothecium. Okay, so in the capsule we are observed that in the Marchantia elaters are present. Some of the cells are going to form the elaters. Okay, endothecium cells act as porogenous tissue. Divisions occur in all planels in these cells and form a mass of 4 mother cells. Okay, from these 4 mother cells, almost half of the cells become sterile and form into elaters. So whereas some cells are going to sterile, so that all are going to form the elaters. Remaining 4 mother cells undergo meiotic division and develop the haploid spores. So this is the sporophytic structure. All are in diploid condition only. Some of the spore mother cells going to meiosis. That after that meiosis, that develop the haploid spores. Initially, the spores appear in tetrads, and as they mature, they slowly separate from one another. So remember this, that elaters are very spindle shaped. It's very important. In the Marchantia, we are observed the sporophyte elaters. Elaters are very important. These are with spiral thickenings and diploid cells. Okay, elaters are spindle shaped structures with spiral thickening and diploid cells. These are hygroscopic in nature. What is the work of elaters? Absorb the water and helps in the spores. This is very important. Elaters. Okay, Marchantia, we are observing elaters. That is very important. And I forgot that in the Archegonia structure, so we are observed that in the Archegonia, that 6 vertical rows of cells in the neck. Okay, so in the archegonial structure, already we are discussed in the general characters class that archegonia and antheridia. Antheridia is club-shaped structure, stalk and body. But in the archegonia, that stalk and venter and neck is there. Okay, so it is a flask-shaped structure. The tube-like neck is with the 6 vertical rows of cells. The neck is occupied 4 neck canal cells. Okay, that is very important. And the upper part is covered by 4 cover cells. The venter is with an egg and ventral canal cell. This remember, Nana, only that 4 neck canal cells is present in the Marchantia. That is remember that. That's very important. The neck is occupied 4 neck canal cells, and upper part is covered by 4 cover cells. Only 4. Okay. So that venter is it is common with an egg cell and ventral canal cells. That is common. That is one of the important part in the archegonia. 4 neck canal cells are present in the archegonia, and whereas we are going to antheridia, so that is different. The number is different. So that's why some chances is there. This is the sporophyte structure. Okay, elaters are very important in sporophyte structure. These are hygroscopic in nature, spindle-shaped structures with spiral thickenings and diploid cells. Remember that. And the next one is the spore dispersal. So after the maturity of the capsule, that haploid spores are developed in the capsule. That capsule matures, that theta elongates and ruptures the calyptra, and pseudoperion brings the capsule out. Whereas these are the wall layers, that is calyptra and pseudoperion. So when the maturity time, that theta elongates and ruptures the calyptra and pseudoperion, it brings the capsule out. The capsule descends into 4 walls. Elaters in the capsule absorb the water and push in the spores out. Okay, when that elaters helps in the absorb the water and push the spores out. So eppadaithe so that pseudoperion and the calyptra are ruptured, so then the time that elaters pushed the spores to outside. The released spores, when they fall on a suitable surface, germinate and develop into a Marchantia gametophytic plant. This is about the sporophyte. Okay, thank you. So any doubts, please ask me.
Okay.
This is the type study of Marchantia.
Any doubt, my dear friends, regarding of today's class? Madam has clearly explained about the hepatic apsida, the type study of this hepatic apsida, that is Marchantia, Marchantials, Marchantiaceae, Marchantia. So Marchantia is one of the very primitive structure, thallus structure. It contains 2 distinct phases. Madam has clearly explained. that is the gametophytic phase and sporophytic phase. In this sporophytic phase, again, Madam has clearly explained about the food seta and the capsule and elaters. The most important terms in this class is the gemma cups and elaters. Gemma cups of the thallus, dorsal side of the thallus, we are observed the gemma cups and gemmas or gemmas, the pulley wheel structure which are useful for the vegetative reproduction, and also the vegetative reproduction along with the gemma cups, the fragmentation we are observed. At the same time, the sexual reproduction, Madam has clearly explained about the Marchantia, the archegonium and antheridia structures, and the sporophyte, the development of the sporophyte from the zygote. The entire sporophyte structure contains the fruit seta and capsule and elaters, and the remaining topic Madam will going to explain tomorrow. And anthoceras, we have the another type study in this bryophyta, that is Anthoceratopsida, the second class. Madam is going to explain tomorrow. So Madam, thank you very much because of you are clearly explained about the Marchantia, and also yesterday you are clearly explained about the general characteristic features of the bryophyta. That is also most important topic. So, my dear friends, regarding of this class, if you have any doubt, Madam is here. Please ask. Otherwise, we'll going to wind up. So, next class sharply start exactly 4 p.m. Now, I will going to wind up the session. Thank you, Madam.
Okay, sir. Thank you. Thank you.